ये मैं भी शुरू बनाना चाहता हूँ।
कर ले फोन उन्होंने पुलिस वाले ने
So welcome everyone streaming for our live sessions. So first confirm me in the chat box if my voice is fine and there is no buffering is happening and you can see everything nicely. Student, please confirm if the streaming is going fine and my voice is also okay. Only then we will start with the lectures. Bache, the resolution you have to Alex make it to 720. So I made it to the highest resolution for all of you. So just try to make it to 720. It will come up very nicely. Okay, good. It seems everything is fine. We can start with our today's lecture. Um, so basically, medicinal biochemistry. There is a one simple biochemistry, and I have tried to input some task to this in order to understand that how it could be benefit from the medicine perspective. So in this course structure of medicinal biochemistry, so today we will discuss very basics of about structure and properties of your proteins and pyrimidines, nucleosides, your nucleotides, structure of DNA and RNA, and we will discuss about central dogma. So some of students who were in my previous class of molecular biology, for them, it might be repetition of the same, but don't worry, this will be the only part. The rest, all the course is different. So be my guest. If you want to listen it again, you can be there. Otherwise, sorry, because I have to, because before I start with anything in my workshop, so this is mainly the 2 day workshop where I have to um, provide you all very basics initially and then go a little advanced. But once we have a this 10 days workshop about these courses, or not 10 days, it's a 25 days workshop, 1 hour each day for 5 days for 1 month. um, so during that time, we go in detail about each lectures, each topic, and study about them in core. So but in these this is like rapid fire question. I have to say we will manage to do most of those stuff that we do in 1 month, but try to finish of that part around 30 to 40%. But still you might be able to get a good grasp of the topics from the lipids to amino acid. I used to teach also virtual labs of DNA isolation, PCR, gel electrophoresis, but I have a Mac Air, but this it has some problem with Flash, Adobe Flash software, so I can't play them. But I will share the links with you. Maybe it's working in your laptop if you're having Windows. So, based on that, now we already had the introductory class. We have discussed this 2 days, 6 hours training program. So, whatever things that I will be speaking here or discussing to you, so I will keep sharing with you in the group so that you and me are on the same order. So currently we are running medicinal, analytical, enzyme, and molecular biology, 4 workshops side by side. So if I speaking of one thing to other, please write me in the chat box. So I will stop speaking about the other workshop. But that will not happen. I will maintain my um, yes, my order. Um, so that was the fun part. Now let's be serious. Uh, Edufabrica, medicinal biochemistry. What? This is not about a teacher. So, as you are all students of biochemistry here, medicinal biochemistry, so I have pretty good collection of what you say biochemistry books. One is Harper, other is Lippincott, and third one I have Lehinger. So I will share all these parts with you so that you could use them for your in future studies. Whenever you have any query, you can refer them, and it will be really beneficial for all of you. So they are like three golden Bibles. So every author has its own specific way of writing, showing diagrams. Uh, Harper is more illustrated on the medicinal point of perspective, whereas the Lippincott is from the college perspective, from the academic perspective, whereas the Lehenga is from the research perspective. If you are doing into some research articles, so I will recommend to go for Lehenga. If you are going for the academic studies to understand to write one answer, subjective answers, then I will suggest you to go with the Lippincott. And if you want to study about medicinal part of biochemistry, then go for the Harper. So each thing has its own importance, own level of complexity. What is this? No. And I also have a little article about liver. Maybe I could also share because it's hanging around with me. Biochemistry and function of the liver, which might be quite essential, quite helpful to the students of who are studying in the medicine or into the biotechnology to core study about liver. All the cycles are there with TCA cycle. This is pretty good. So I must share. I've never seen that before. My eyes just went on it today. So you can also have this one. So I will keep sharing the things, the PPTs, the books, whatever the material that I will having onto this link. You can pin them down for yourself um, to come up over there and watch them anytime. So the uploading is very slow at the moment. Okay, uh, and also all students, please make it to 900 today. I have now 897 subscribers. So this is all because of the online coaching at the moment, not coaching, workshops that we are going through. So make it happen to 900 today. It will be really helpful. Um, and if you want to go for another workshops, you can go. But I will say, um, yeah, okay. Let's start with the nucleic acid part first. So at the moment, this is not your basics of molecular biology class. This is just to explain the first topic of your nucleic acids, to differentiate between your nucleosides and nucleotides in order to understand DNA and RNA. We will just base our studies on that at the moment. Then we will switch to another topic later on. So let's start with the eukaryotes and prokaryotes. As we have discussed, that the eukaryotes are present in mostly in your plants, animals, and fungi, whereas the prokaryotes, they are present in the bacteria and cyanobacteria, the primitive ones. And the cell wall in the eukaryotes, it's not present, and in the plants, it's present. Whereas the prokaryotes has a yes, they are present. The cell walls are present. Beyond that, meiosis is not happening in prokaryotes, whereas in eukaryotes it's happening. And the ribosome size is around 80s in the eukaryotes, and then prokaryotes 70s. And all these cell organelles, like important organelles, like your nuclear membrane, endoplasmic reticulum, Golgi apparatus, mitochondria, chloroplast. They're all present in the eukaryotes, whereas they're all absent in your prokaryotes. So that's the basic difference here, eukaryotes and prokaryotes. So now let's go to the basic strong foundation of our body, that is a gene, or we can say which plays a big role in order to understand things. So first of all, um, so I need to switch off this part. This is not required. Here, from here we can start. That is your gene, which is a unit of our heredity units. That segments that carries genetic information that are called genes. So how these DNA, this amount of DNA is carrying out all the information into the chromosome, and how all the information has been compiled, that's one of the interesting things that keep us always surprising. Yeah. So in order to understand that, so let's see this diagram. So here in this diagram, you can see that we having our DNA double helix, which is of 2 nanometer diameter. So at this is at the very simplest level of chromatin, just double helix structure we can see. Then further, it's getting having a 1.65 turnover over these histones molecules, which are made up of 8 histones, and wrap around them 1.65 times. And also there is some like a switch on side with H1 histone, which helps to chromatosome consist of H1 histones here. So this is your DNA, your histone, and your H1 together. It's known as your chromatosome, which is of 11 nanometer height. Then these beads on a string, this structure is also called as beads on a string. They get folded together, and they make a bunch of balls and around 330 nanometer of height. And then further they get folded to form loops averaging of 300 nanometer length, making to a zigzag shape like this. You can see this zigzag shape with 250 nanometer wider. And then these wide 250 nanometer wide fibers, and in the length around 700 nanometers, they also get again zigzag, get compressed even more deeply, and they become the sister chromatids of your chromosome. These are smaller sister chromatids, the bigger one, and the total height is now 1400 nanometer. So they're starting from 2 nanometer to 1400 nanometer, and tight calling make this your chromosome. So we can see how nicely our DNA is packed in our body, and how things are being um, good to go for body. So that's the basics of our DNA. That is how it is coiled around your chromosome. I will share this image, and the image that we see is quite blurred there, so this is the right image for the same. So here we can see also that cell is there, then the chromosome. In the cell we have 23 pairs of chromosomes, and chromosome is made up of genes revolve around these beads, and the gene consists of DNA, which is made up of 4 chemical letters A, C, T, G. So they will slowly and slowly being uploaded, so no worries. So yeah, DNA, which is a deoxyribonucleic acids, which contains the genetic instructions used in the development and functioning of all living organisms, and some viruses also. So they act as like a blueprint for your proteins and RNA, all other molecules. So quite important part playing role as DNA. So how does the structure looks like? The structure is a double helix structures having all your basics foundations of A, T, C, G, purines and pyrimidines pairing with each others. Purines are pairing with the pyrimidines. So A is pairing with the T, and then G is pairing with the C. And while pairing A and T, they are making a double hydrogen bond, whereas G and C, they are making triple hydrogen bond. And because of this bonding, this DNA has a double helix structure, and this maintain its stability. And beyond that, we having this deoxyribose sugar on side chain with these nucleo nitrogen base pairs, and they are these phosphate groups are being attached with each other with the help of these sugar groups are attached with the help of phosphate groups here. And on the one side, it starts from the 5' end and it's ends at the 3' end on the left side, and in the right hand side we have a 3' end to the 5' end on the right hand side. So this is a DNA double helix structure. So there are two strands that run in opposite directions to each other, and therefore it's known as a anti-parallel structure. So chemically, DNA consists of two long polymers of simple units called nucleotides with backbones made of base, sugar and phosphate groups. And what is the difference between nucleosides and nucleotide? Basically, your nucleoside includes your sugar plus your nitrogenous base pair, whereas in the nucleotide it includes both sugar and base pairs are there, but it also has phosphate group. Yeah, sugar plus base include making a nucleoside, whereas the phosphate plus sugar plus base together they make your nucleotide. So that's the major difference between each of them. And what are the purines and pyrimidines, and how their structures differentiate from each other? Mainly due to the membrane ring. So the purines are 5 and 6 membrane heterocyclic compounds, whereas the pyrimidines are only 6 membrane rings are present. So in adenine, so the example of purines are adenine and guanine, whereas the example of the pyrimidines are cytosine and thymine, that is your 6 membered rings. So further in the adenine and guanine, what we can see that it's a 5-membered ring here and 6-membered together as adenine, and this is guanine. But there is a difference of between thymine and uracil of methyl group, and uracil is mainly present in your RNA and thymine is present in your DNA. And the pairing, as we have discussed, A pairs with T in the double bond and A pairs with the U on the double bond, and the G on the other side pairs with C in the triple bond. So here we can see the bonding between adenine and thymine, that is a double bond between two nitrogenous base pairs, and in the guanine and cytosine they are making a triple hydrogen bond. Further, we can see that the difference between your both ribose sugar in DNA and RNA is that the main difference is the second carbon, which is a hydroxyl group, is not present in the deoxyribose in DNA, whereas in RNA in the second carbon on this ribose sugar, hydroxyl group is present. That's why it's a ribonucleic acid. And also in this DNA, remember that we have minor groove and major groove. Though these things seems to be very, theoretical, I'm just explaining you class 12th molecular basis of inheritance. If you open your NCRT, these all figures are there, and even the class 12 student, once he will read all these, he could explain us. So I'm not doing anything something new. I'm just repeating our past here, our basic things. But my point here is for all of you to let you think that all these findings by Watson and Crick in 1955, when they were doing their research, it was not a single day work. So they have to go through X-ray crystallography of many structures in order to be successful with this structure, and they got also a Nobel Prize winner for the same. So these things take a lot of time, lot of pain, lot of frustrations, a lot of your weekends off in order to find the results for the things. So just remember, so whatever the discovery that you are reading, whatever the thoughts that you are reading, so you can understand that a lot of researchers, lot of scientists, hard work has been lying there for the same. Whether you are yeah, or any enzyme that you are studying or any drug discovery you are doing, don't think that thing has been done in a single day. Or your paracetamol, crocin, this COVID vaccine that has been produced with the AstraZeneca Covishield in India that we are having and being produced in mass production by Bharat Serum. So this was not, you know, a one-day work. It's a lot of money has been input in there. A lot of scientists has been working on that day and night. and in order to, yeah, protect us. So ribonucleic acids, next is your RNA, which is a biological important type of molecule that consists of long chain of nucleotide units, and these contains also your nitrogenous base, ribose sugar, and phosphate group. So it's not like double helix structure always. So it's also sometimes single helix, single chain, and then it's become double chain a little bit for few base pairs. Then again becomes single chain, and then again it's become double and then single. So like this, your RNA could be like this. Student, just give me a minute. I'm coming back just for emergency. Just a second. Okay, just a second. So this your DNA could your RNA could be also be double stranded. It's happened sometimes in viruses. They mostly try to have a double stranded RNA in them. Now let's talk about types of RNA. Basically, they are messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA includes your codes for proteins, whereas ribosomal and tRNA is mainly used for your translation. And on the other hand, in the post-translational modifications, your smaller nuclear RNA, which is used for splicing, and yRNA and telomerase for the all post-transcriptional modifications such as RNA processing and telomere synthesis respectively. Beyond that, if we discuss about messenger RNA, which carries your information from your DNA to the ribosomes for the protein synthesis. So it's act as a messenger. So that's why it's known as a messenger RNA. And it is coded so that every three nucleotides correspond to one amino acids. So like GUU, ACC, GAU, like this. So and mostly during this process the introns are removed mainly and exons are existing during the RNA splicing and transcription. So here we can see your DNA got unwinded, whereas a coding DNA will produce your messenger RNA with the non-coding strands being removed on the side. So messenger RNA is then exported to nucleus to cytoplasm, where it is bound to ribosomes and translated to its corresponding proteins, which form the with the help of your tRNA. So this statement that I just a second. Oh my god. That mRNA is then exported from nucleus to cytoplasm, where it is bounded to ribosomes and translated into its corresponding protein to form with the help of tRNA. So, in order to discuss this thing, let's switch to now um, some interesting videos about that. Before we start some interesting videos about transcription and translation, I just want to show you something that makes me even still think of the cell size and scale. Yeah. So if you see here is your coffee bean, which is the size of 12 to 8 millimeter, and this is a grain of rice, which is of size of 8.8 into 2.5 millimeters. The sesame seed, that is your till, 3 by 2. Grain of salt, 0.5 millimeter. Then comes your amoeba. We go into biological part now. 500 micrometer, the micrometer parts of your prokaryotes. Then the paramecium, 210 by 60. Now here the human egg is size of 130 micrometer. In our eyes, photoreceptors rods about 2.5 to 100 micrometer. Then sperm cell is around 60 into by 5, 60 by 5 micrometer. Skin cells of around 30 micrometer. Now we go more detail into our cell, that is your red blood cells around 8 micrometer. So we have discussed about mostly all your prokaryotes, human cell skulls. So how they are getting smaller and smaller. Now let's go even deeper. That is our X chromosome. This is size of 7 micrometer. Baker yeast is around 303 to by 4 micrometers. So everything we are discussing is in micrometers. So now let's go even further deep. Bacterium, E. coli, 3 by 0.6 micrometer. Lysosomes 1, mitochondria 4 by 0.8. And then even the viruses, now they go into the nanometers, as we know, they in the nanoparticles. So they go for measles virus around 220 nanometer, influenza virus around 130 nanometer, HIV around 130, your phage bacteria virus around 70 by 200 nanometers, hepatitis 45, rhinovirus 30. So these COVID virus, all these are in nanometers. Now comes the that what we were discussing, ribosomes and tRNA and mRNA. So they are also in nanometers. So like around 30 nanometers. Your tRNA is around 7 nanometers. Your antibody is around 12 nanometers. You know, hemoglobin 6.5 nanometers, even smaller. Now we go even deeper. So here we can see our nitrogenous base pairs, adenine, glucose, methionine. They are in picometers now, even smaller than the nanometers, picometers. That is your 10 raised to power minus 12. At the end, water molecule 275 picometer and the carbon atom around 340 picometer. So starting from carbon atom here, that we say mostly the reason for life or the first molecules that were present in our atmosphere, carbon, oxygen, all these. So methionine. So we can see that how from picometer to nanometer to micrometer, millimeter times regular 12 point that we use in order to write on our WordPad. Um, how everything is seems to be connected over the size. So this make us to think that how things are tiny that even we can't think with the naked eyes, but they're still present, and we have that technology that we are able to see them, watch them, and understand them at a bigger level. So share this. Excellent connection, that's good. So now next, what are DNA and genes? So let's have a look over this.
The instructions for building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order. But small differences in the sequence of the bases in our genes make each person unique.
So all these differences in the nitrogenous base pairs make us different. Now let's see what are proteins.
Proteins carry out most of the body's functions, and they make up the bulk of its structure, from hair and fingernails to bone and muscle. In fact, nearly everything in our bodies is made from or by proteins. The instructions for building proteins are found in segments of DNA called genes. Proteins are made of long chains of building blocks called amino acids. There are 20 amino acids in all. Put together in different combinations, amino acids form proteins with a broad variety of shapes. This diversity of form gives proteins their many functions. The proteins a cell makes determine what it can do. Our cells are constantly at work, producing the thousands of different proteins that keep our bodies functioning.
So that was about protein. So yeah, just share with you this one. So this is very nice website where we can understand the basic things about DNA, RNA, biochemistry. So let's discuss about same gene, different organisms. How having a same gene, but we can put it into different organism like yeast or into the fishes. So they give us the same thing that we are looking for, like jellyfishes, who have a tendency to glow in the dark, and we got their gene and we put them in the fish, and they even started to glow in the dark. Or we take some spider web from the gene from the spider web, and we grow them with the help of yeast, and then we have the overproduction of this enzyme for special use. So let's discuss this video about the same.
These may look like ordinary fish. What you can't see is that they have a gene in them that codes for green fluorescent protein. But shine a blue light on it, and GFP emits a bright green glow. What might surprise you is that the gene for GFP came from a jellyfish. GFP was first discovered in jellyfish over 50 years ago. Since then, scientists have been using it to study all kinds of biological processes. What's amazing is that this gene from jellyfish can be put into just about any living thing: animals, fungi, plants, bacteria. They can read the DNA code of a gene and and build GFP that is identical to the jellyfish protein. That same concept applies to tons of other genes too. The gene that spiders use to make silk webs, for example, can be put into yeast cells. Harvest the silk protein that the yeast cells make, and you've got the raw material to make a strong, lightweight fiber. In fact, a company is using this approach to bring yeast-grown spider silk fabrics to market. How is it then that fish and yeast can read DNA from a completely different organism? It's because, despite their differences, there's something fundamentally the same about how all living things read genes to build proteins.
I hope all these things making some sense to you, all of you who are present in the session at the moment. Now, last thing about let's talk a little bit about transcription and translation.
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life. Transcription and translation are the processes.
Just a second.
Transcription and translation.
So we can take a gene here.
Inside the nucleus, RNA polymerase attaches to a gene. It separates the two complementary DNA strands, exposing the gene's unique sequence of bases.
So T will pair with which one? A, G, U, or C? It will pair with A. G will pair with C. T will pair with U. Sorry, A. Sorry, is with the C. Is a late night effect. A with U, C with G, T with A. So like this, it's continue.
When the end of the gene is reached, transcription is complete. The new strand of mRNA passes out of the nucleus and into the cytoplasm, where translation takes place. The instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated, one after another, in order to build a functioning new protein. The key structures in that process are the ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon and a matching amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with a ribosome. The ribosome attaches to the end of the mRNA strand. It slides along until it finds a start codon, AUG. Through complementary base pairing, the start codon pairs with the anticodon on the tRNA molecule, signaling the beginning of the protein instructions. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand, one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. Give translation a try.
So let's see, as we have discussed now, that how with the help of ribosomes and your tRNA, these codons on mRNA, anticodons of tRNA will be correspondingly attaching to the mRNA's codon. So like for ACU, it will be UGA. Where is UGA? So it will be threonine amino acid will come on top. So like this, the cycle will keep going on, UGU. So for that, it will be ACU, ACA, sorry, cysteine. So like this, cycle will continue.
When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acids folds into its final form and the new protein is complete. This protein, aminoacyl tRNA synthetase, will now attach the right amino acid to the proper tRNA molecule, charging the tRNA so that it can participate in protein synthesis. Each type of amino acid has its own enzyme. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life.
So I hope this transcription and translation process might have been cleared forever in a very easy peasy manner for all of you. Okay, now we continue with our slides. So in prokaryotic cells, cells which do not have nucleus and cytoplasm compartments, they can bind to ribosomes while it is being uh, transcribed from DNA. Then transfer RNA, we will just have a little bit view about them. The transfer RNA, as we have discussed, it helps in order to produce your amino acids. Ribosomal RNA have two types, 70S and 80S, whereas 70S includes your 50 and 30, where 80s includes your 60 and 40S. And the difference between your DNA and RNA is basically is that RNA's nucleotides contains ribose sugar, whereas the DNA contains deoxyribose. RNA has a uracil that has a thymine. There's a presence of a hydroxyl group at second position of ribose sugar. On DNA, it's absence. RNA is usually single stranded. DNA is a double stranded one. I think we should stop here. I want to teach you about DNA and RNA here. So if we see from your syllabus perspective, so we just have to discuss about structures and properties of purines and pyrimidines, nucleosides, nucleotides, structure of DNA and RNA. Let's discuss a little bit about central dogma, because without discussing proteins, we can't come up to the level of your central dogma here. So in order to complete that part of your nucleic acids, so we will also discuss a little bit about proteins. So basically proteins are also known as polypeptides that are made up of amino acids arranged in the linear chains folded together into globular form. And the sequence of amino acids in a protein is defined by the sequence of a gene which is encoded in the genetic code. And genetic code that specifies us 20 stranded amino acids in them. So here we can see a very nice structures where cysteines are making disulfide bridges. and all other amino acids are being playing role here. And also we see these together making a quaternary structure which could be present in your pulses, um, red kidney bean, peanuts, chickpea, fishes, meat, chicken breast. So all these things have an ample amount of protein, and it's quite essential also. So what are the basic players that are playing in the molecular biology, as we have discussed? Mainly DNA, RNA, and proteins, and how they work. So mainly DNA could also replicate, leads to the production of your RNA with the help of transcription, and RNA in the process of translation produce your protein. And this whole thing, as we have discussed in the video also, is known as central dogma, that is center to the life. Okay, it's there we can discuss that DNA replication is the basis of our biological inheritance, and it's a fundamental process occurring in all living organisms to copy their DNA. And this happens, just here we can see in the structure that first your DNA is winded up. It's a double helix structure, so it gets unwinded with the help of topoisomerase enzyme, and then helicase enzyme will separate your nucleotides, leading to the production of lagging strands and leading strands. आपके lagging strand and leading strand आ जाते हैं. The leading strands will make a new your polymerase with the help of DNA polymerase make a corresponding template, whereas with the on the other side it will make some Okazaki fragments, even though there some breakage between these opposite template, and this will be removed by RNA polymerase and DNA primase, and they could be joined also by DNA ligase. So DNA ligase will come and fix the broken part in this. So we had discussed transcription, that is your DNA to RNA, and if it is reversed, then it's a reverse transcription. During the disruption, RNA complement includes uracil instead of thymine, and this is a process in which messenger RNA will move from your nucleus after getting the message from the DNA to the cytoplasm part. So DNA will be read from the 3' to 5' end during the transcription, and the complementary RNA that is produced will be from 5' to 3' end. So only one of the two DNA strands called the template strand is used. Mainly one strand will be used for this, and which will lead to the production of single-stranded RNA like this. So here we can see some hairpin loop also being made, this circular one, and your RNA polymerase is removed afterwards. The function of it is done. Also, there is a RNA synthesis and processing, that is in the transcription. You have your introns 1 and intron 2. So these introns 1 and 2 of the gene will be excised, will be removed during the RNA splicing time. Once it is done, it is removed, you will have only messenger RNA consist of exons which are coding sequence. So that's happened during your RNA synthesis and processing. That's reverse transcription. If you HIV RNA with the help of reverse transcriptase, it could produce your HIV DNA also. So that's the basic central dogma here, what we are seeing here. That is the blue one are the general case, that is that could happen in normal cases. The DNA could self-replicate, then RNA could produce from that, and then protein could be produced. But there are some special cases that RNA could self-replicate, and then RNA could also produce complementary DNA with the help of your reverse transcriptase. So these are the special cases according to the central dogma given by James Watson and Crick. So this is the end part of this topic, that is your translation. So if we are discussing about our all the basics, nucleotides, nucleosides, DNA, RNA, central dogma. So translations need to be also discussed in that. So basically translation is divided into three parts, that is your initiation, then elongation, then termination. So in the initiation part, your methionine consisting of tRNA will join with our start codon with the help of ribosomes over the messenger RNA, and then continuously various other tRNA will keep coming and giving up as a complete chain of a polypeptide chain of amino acids. That what we have discussed just now. That what we have just discussed now in the in that video also. And what is this genetic code is made up of? Basically, your genetic code, you can create this table very easily. Just make your UCAG on the left, and on the top make UCAG. So it says 4 by 4 box, 16 boxes will be there. And then on the right hand side, UCAG. Yeah. So UCAG will be all consisted here. So from the first, if you want to make the first one, you will have the first base pair U. From the second base pair, you will have this blue one, and the third base pair, the first letter from the top letter U. So like this, four base pairs, like all these things will come together, and we will write the letters. So the one we see the yellow one, so they were complete set of amino acids. So these are called valine, leucine, proline, serine, alanine, glycine, arginine, like this. Whereas the together, and also remember this methionine, which is a start codon AUG, and the stop codons are UAA, UAG, and UGA. These three are your basically your stop codons. So that was about the genetic code, how it works. We have discussed basically about DNA and RNA in complete sense in today's lecture. So I will not go to this genome part, into this genome analysis. Neither we are going for any tools, gel electrophoresis, because they might be a bit out of the focus for today's lecture. So why this one is not being sent? Okay, no problem. So students, just give me 5 minutes. I will be back. Yeah, I will be back within 5 minutes, and then we start with the next topic of biochemistry. So we will now go with the hardcore biochemistry. I'm just coming within 5 minutes. Need a little throat rest. So, students, welcome back. So, please subscribe to the channel also, students. It will really help me. Oh, 900. Woohoo! Thank you so much. Thank you so much. Now it's 1000 it has to achieve. This is, our life is everlasting, fighting for more and more. Our hunger will never be over. Um, so something now we will go into more part of biochemistry now. So we have discussed our nucleotides, nucleosides. Uh, so let's discuss some structural behaviors that are in the cells, tissues, and whole organisms. We will discuss now, from now onwards, about structure and behavior of the complex molecules found in biological materials and the way these molecules interact to form cells, tissues, and whole organism. So, what is actually biochemistry is all about? It's actually applications of chemistry to study the biological process at the cellular and molecular level. So, it is emerged as a distinct, a totally different field by the beginning of 20th century when all the scientists combined your chemistry, physiology, and biology in order to investigate the chemistry of living organisms. And on the other side, if we are focusing on the medicinal biochemistry, that is more focused on to find out the diagnosis of your various diseases, whether we are doing LFT, KFT, jaundice test, like we are doing this RT-PCR test. All these things is behind is your biochemistry. So in order to apply biochemistry role for the application of medicines, that term came out to be as medicinal biochemistry. So some principles. So cell, which is a basic structural unit of living organisms, they are highly organized and they provide constant source of energy, which is in order to maintain the ordered state of our body. And then living process contain thousands of chemical pathways, precise regulations and integrations of these pathways are required to maintain the life. And certain important pathways like glycolysis is found in almost all organisms. And all organisms use the same type of molecules like carbohydrates, proteins, lipids, and nucleic acids in order to survive. So instructions for growth, reproductions, and development for each organism is encoded in their DNA. So that's these 5 points that I had just spoke within 10 seconds. I have to say, but believe me, these five points have lead to writers lots of books. A lot of research has been gone, and these things are very summarized of everything. So basically, your biochemistry includes certain pathways, glycolysis, TCA cycle, Krebs cycle, photosynthesis, all these cycles, all these important pathways, which is almost found in all organisms. And we also see same types of molecules in our body, like carbohydrates, proteins, lipids, and nucleic acids. And beyond that, in order to, what should be the method in order to grow our body, in order to reproduce, in order to develop in each organisms, that is all information as we have discussed. Gene as a hereditary unit, that is your DNA has it, yeah. And beyond, furthermore, I want to add to that, that is living processes contains thousands of chemical pathways, precise regulations and integrations of these pathways required to maintain the life. Meant कि आपके जितने भी these as we have discussed chemical pathways, and they are all regulated, like which enzyme need to be produced at what time, and which things need to be isomerized, which need thing to be oxidized, which thing needs to be reduced, when thing has to be hydrolyzed, when thing needs to be dehydrolyzed, when things needs to be phosphorylated, when things need to be dephosphorylated. So all these complex systems that are happening in our body, itself is very curious, and we doesn't know how many other methods are being governed in our body. So thing is, being a scientist, being a researcher, what you are doing is you are finding the already things have been found out. What you are doing is, as a scientist, DNA was already there. Yeah, we know that there are bacteria. We already know that there's a COVID virus also. We already know everything is already present. We are just the one who, as a human being, with our own intentions, with our own level of intellect, we are able to discover these things and able to tell to the world that these are the things that are working at particular level. So that's the point of being as a scientist or researcher is. So we are just discovering the undiscovered things in the world in order to let the people know, okay, this is how this mechanism is was working. This was working also before as by the nature, but if we interfere in this pathway or something, so we could get something out of that also. So that's what we are doing since, I don't know, BC. Like, we are producing with the help of your biotechnology, cheese production, wine production, all these things, bread making. These are all things that we are using, actually harnessing microbes in order to produce things which are more tastier, more healthier, and more wiser. Now, cell as a basic structural unit of a living organism, that's the basic thing that we are discussing here, that how cells keep making ATP on a continuous level, how it's keep maintaining the nice level of energy during the whole process. So students, if you have any questions, please do write it here. I will be happy to reply you in order to answer your questions. So feel free to do so. So now, cell, which is your basic building block of the life, like if we are making a building, in a building, bricks are the basic building block of the building, and this, yes, they're the smallest living unit of organism also, and they grow, they reproduce, they use energy, they adapt, and they respond to the environment at the end. So many cannot see them with our naked eye, so we need some microscope in order to see them. And as we have discussed cell size with the microbes, yeast, viruses, we have discussed all these things. So everything make a sense here. So cell may be an entire organisms, or it may be one of the billions of cells that make up the organisms. And these are the basic types of cells. So here on the left side we can see a eukaryotic cell. On the B side, on the right hand side, we can see a prokaryotic cell. So, as we have discussed in our first lecture of today in the basics of molecular biology, so we have discussed the cell organelles are mostly absent in the prokaryotes, and most of the cell organelles are present in your eukaryotes. So here we can see nucleoid region contains the DNA in prokaryotes, cell membrane and cell wall is present and contains ribosomes to make proteins their cytoplasm. Where on the eukaryotes, it has three basic cell structures, that is your nucleus, cell membrane, and cytoplasm with various cell organelles. You like Golgi complex, nuclear envelope, chromatin, nucleolus, centriole, SCR, RER, microtubules, mitochondria. So all are being gathered nicely. Now, essential thing in biochemistry we are studying is the water, which is about 60 to 90% of organism in water in our body, and it is being used in most of the reactions in our body also. So that's why it's called as universal solvent. So when you are doing any research, extracrystallography of a protein structure, so we will see a lot of hydrogen molecules being attached to it. So the reason behind it that because if they are present in our body, because our body is around 70% of water, so all the proteins that will be working in the body, they will be having hydrogen molecules around them also. So that's why water is always essential in extracrystallography or any other things that you're working for. Then comes biomolecules. Just like cells are building blocks of tissues, likewise molecules are also building blocks of cells. Animal and plant cells contain approximately 10,000 kinds of molecules. and water constitutes around 50 to 95% of cells content by weight. So ions like sodium, potassium, calcium, they may account for another 1%. And almost all other kinds of biomolecules are organic, that is CHNOPS, and we can have infinite variety of molecules that contain carbon in them. And most biomolecules considered to be derived from hydrocarbons, and they are known for the organic biomolecules that are determined by the functional groups, and most biomolecules have more than one functional unit. So if we see the building block, they are the macromolecules. Simple sugars are the polysaccharides. Amino acids, they are made up of proteins, and nucleotides, they are made up of RNA and DNA, and fatty acids, they are made up of lipids. So for these building blocks and the macromolecules, we can see simple sugars, they are making polysaccharides. Amino acids, they are making protein. Nucleotides, they are making your RNA and DNA, and fatty acids together, they are making your lipid. So this diagram is very actually, I want to make a heart here, because this have summarized the biochemistry. This have really summarized the biochemistry. All your things will be interlinked. Everything, your lipids, DNA, protein, polysaccharides, all things will be making sense here. So if the macromolecules are being produced to the building blocks, then it's a catabolic. But if from building block to macromolecules, it's known as an anabolic reaction. I think we should stop here today, so I can have some question answers from all of you students. If you have any questions, you feel free to ask me. I will be happy to answer them. And did I share these presentations with you already? No. So I will share this molecular biology presentation. Introduction of biochemistry. I hope I have touched you basic for today class. So we will discuss today about biomolecules, water, cells. Yeah, we have discussed about the basics of principles of biochemistry. What is biochemistry is about. So we have touched quite number of topics related to biochemistry. And before that, in the today's evening lecture, we have discussed about DNA, RNA, transcription, translation, various types of RNA, and how protein synthesis is being done. And then we saw many virtual videos regarding the same in order to understand the concept very well. Okay, for our today's lecture, and for tomorrow we will see us at the same time, same place, 8 p.m., 8 to 10, and we will continue like the same. So I'm open to your questions. If we have no questions, we can make the session done, because last 10 minutes are meant for the sessions. Or anything you want to ask about your future perspective, or where should you go, how should things should work, or any personal things, I will be happy to answer those questions also. And you're most welcome to ask me anything, um, any query that you have. Okay, I couldn't hear anything from any students so far, so I will end the session here. Then take care, students. We see us tomorrow, same time, same place. Stay safe, yeah. Bye bye.